Welcome, everybody. I'm really delighted to present Dr. Alejandro Sweet-Cordero for today's special seminar. He gave a distinguished lecture presentation at noon today that was recorded, and I know most everyone was in class at that time, so you can check that out. I listened in on Zoom, and it was really good, and it's very relevant for everybody here as well. So for this afternoon session, the topic is drivers of therapy response and epigenetic plasticity in osteosarcoma. So he's going to talk about his own laboratory research. Dr. Sweet-Cordero got his MD at UCSF and then went to Dana-Farber for residency in pediatrics and then a fellowship in pediatric oncology. And now is back at UCSF as a professor in pediatrics and chief of pediatric oncology and director of the Molecular Oncology Initiative, which is what he talked about at noon today, really all of the molecular analysis they're doing of their cancer patients and sequencing and informing treatment decisions. So that was really interesting. So very well published, well funded. We're really grateful to have you here today. And would your other questions get held to the end or any time? Okay. Okay. All right. Thank you.
Great. Okay. Everybody hear me? Great. Well, thank you for that kind introduction. And thanks for coming. I know this is not the usual seminar hour, so you probably were all in the middle of doing experiments. So I appreciate you taking the time. What I wanted to talk about today is some work that we're doing in my lab to study osteosarcoma, which is a rare cancer, although not so rare if you happen to walk through a pediatric oncology inpatient ward. So I'm going to talk about today is a little bit of introduction to the clinical features of the disease and why I think it's an interesting disease to study from a point of view of cancer biology. Give a little primer on bone development because I think that's helpful to understand when you think about osteosarcoma. And then talk about our work to study the osteosarcoma genome and the epigenetics of osteosarcoma. And I probably won't get to the single cell analysis, but we can talk about that offline. My lab has been studying osteosarcoma for quite some time now. We have a number of projects related to osteosarcoma that I won't really have time to get into today. We're very interested in identifying combination therapies. We're very interested in studying metastatic drivers, developing genetically engineered mouse models, and studying the connection between DNA damage and innate immunity. But most of those topics I'll have to skip today for lack of time. I wanted to just start by sort of a fun factoid, which is that osteosarcoma is probably the most ancient cancer known to science. This is from a nice paper that was published a couple of years ago from a dinosaur that was shown to have osteosarcoma. And the relevance of that today is that actually, in addition to humans, large dogs also get osteosarcoma quite frequently and are a nice companion system to study osteosarcoma. So we have a lot of colleagues on the veterinary side that also study osteosarcoma. So this is, as I said, it's a rare cancer. There's about 900 new cases every year in the US. The survival is actually pretty good if you're localized and if you don't have metastatic spread at diagnosis. With surgery and neoadjuvant chemotherapy, you can be cured about 60–70% of the time. Unfortunately, a lot of patients present with metastasis, and when the disease is metastatic, it's much harder to treat. And one of the things that motivated us to study osteosarcoma in the lab is that unlike a lot of other cancers that you're all maybe more familiar with, including skin cancer and others where there's been a dramatic improvement in access to targeted therapies, osteosarcoma is really essentially treated the same way today that it was treated 40 years ago, which is intensive chemotherapy with a combination of methotrexate, cisplatin, and adriamycin, and that really hasn't been much improvement in that. And from a point of view of cancer biology, it's a very interesting cancer because unlike a lot of adult epithelial cancers where there's a known genetic driver, the characteristic feature of osteosarcoma is this incredibly complex genome that you can see reflected here in this Surcos plot with the lines in the middle indicate structural rearrangements. If you look at the single cell level, this is a collaboration we did with a expert in sky analysis at University of Minneapolis, Betsy Hirsch. This is one of the cell lines that we had generated from one of our patients. And you can just see each chromosome is painted a different color. There's just an incredible amount of aneuploidy, chromosomal rearrangement, chromothripsis, and other complex genetic features that I'll talk a little bit more. We've also done quite a bit of work using RNA-seq to characterize bulk RNA-seq to characterize these tumors, and this just indicates the heat map here that there's a lot of heterogeneity in the tumor microenvironment. So in addition to the tumors being heterogeneous from one to the other, there's a lot of intratumoral heterogeneity. There's also a lot of heterogeneity in the tumor microenvironment. So backing up a little bit, just as a reminder, you probably all studied this in college or med school at some point, but bone development is actually quite interesting and quite complicated. So there's on the right side here, you see the osteoprogenitor cell, which gives rise to preosteoblast. And then either on the inner surface of the bone, on the bone marrow surface, or on the outer surface of the bone, there's this layer of cells called osteoblasts, which basically generate the final mature bone cell called an osteocyte, which then produces the bone matrix around itself and sort of becomes encased in that matrix. Osteoblasts are thought to last for a few weeks. Osteocytes actually last for years, and are probably one of the more stable cells in the body. In addition to that, there's also an important role for hematopoietic-derived cells, which give rise to macrophages, which can then differentiate into osteoclasts, which play a really critical role in remodeling of the bone. And normal bone development has been quite extensively studied over many years, and it's well known that there are very well described transcription factors that have to be activated during bone development. So on the left here, you can see SOX9, which is turned on early during bone development. Then a little bit later on, RUNX2 is turned on. RUNX2 then needs to be turned off so that an osteoblast can differentiate into a mature osteocyte. And these transcription factor master regulators are in turn driven by a series of very important signaling pathways illustrated on the top of this graph, including the GLE pathway, Notch signaling, beta-catenin signaling. Again, each one of these is turned on and turned off in very specific developmental timing. How those different pathways are related to osteosarcoma development is something that is not totally understood, but I'll touch on that, on how we think it's implied in some of the research we've done a little bit later. Just from a histological point of view, there's also quite a bit of heterogeneity in osteosarcoma, which to some extent reflects this different stages in bone development and may suggest that different osteosarcomas are initiated from different cells in a continuum of bone development. So on the left here, you see a so-called osteoblastic osteosarcoma, which still makes a lot of this bone matrix, which is this kind of pink stuff that you see here, so-called malignant osteoid. There's also a chondroblastic form of osteosarcoma, which makes more of a cartilage matrix, which again may suggest a different cell of origin. And then there are some osteosarcomas that don't really make matrix anymore, and they're so-called fibroblastic. So this kind of primer on bone development is helpful, I think, and it gives us a lens on through which to see the development of osteosarcoma and ask critical questions that may be important for understanding the development of this disease. So, for example, at what stage of bone development does osteosarcoma occur? To what extent are the transcription factors and signaling pathways that are important for bone development, also important for development of the cancer. How do these peculiarities of bone as a tissue, for example, the fact that there's a hard matrix, and a lot of times osteosarcomas are also encased in this hard matrix, how does this influence the evolution of this genomic catastrophe? So, for example, is it possible that some of the sort of high tension and high matrix, high tense characteristics of the matrix may impinge on the development of osteosarcoma and cause it to have additional damage to the chromosomes or development of micronuclei, which then lead to development of chromothripsis and other forms of structural rearrangement? How does it also regulate anti-tumor immunity? And how does the close proximity and crosstalk with the hematopoietic system that occurs on the inner surface of the bone influence the development of osteosarcoma? So these are all questions that we're interested in studying in the lab. At the end of the day, though, this is really the clinical process that we're trying to understand and inhibit. As I mentioned earlier, if the disease is localized, the diagnosis, and it can be cured. If it's metastatic, then it becomes very difficult to cure. So this is actually a patient of mine that I took care of a few years ago who had the CT scan shown here on your left at the end of treatment. And then about a year later, we scanned her. We were scanning her every 3 months, and between one scan and the next, she came back with this very large right posterior metastasis. And when you see that, then it's a completely different disease, very difficult to treat. You can try to resect that metastasis, but the outcome is generally not very good. So what we're trying to prevent is, ultimately is the development of metastasis, or at least figure out how to better treat it. We think a lot of patients probably have metastasized already at the time of diagnosis, but for some reason, some patients' micrometastasis are eliminated by chemotherapy, whereas others are not. And so understanding that is also something we're very interested in. I mentioned at the beginning that there's been lots of efforts to try to improve therapy, but that we're still using the same therapy that we used 40 years ago. This is a slide from a very disappointing, very large study that was done in collaboration with the European groups, published a few years ago, where they tried to improve on the current methotrexate, adriamycin, cisplatin treatment by adding more chemotoxic chemotherapy, cytotoxic chemotherapy, in patients that were deemed to be high risk. So patients that had poor necrosis, that surgical resection, were given an addition of, in addition to MAP, they were given ifosfamide and etoposide, and essentially made no difference. So they just got a lot more toxicity, a lot more time in the hospital, but really made no difference in their long-term outcome. So the approach that we've taken in the lab now for several years and to try to study this disease, one of the problems that we encountered was that there were not a lot of great models. So there's some cell lines. If you go to ATCC, you can buy, U2RS cells, you can buy SJSA, these cell lines that have been around for 40 years that are well known to many cancer biologists. But to what extent they really reflect the heterogeneity of the disease that I mentioned to you earlier was not clear. So we embarked on this, rather technically challenging and logistically challenging effort to try to collect as many metastasis and primary tumors from patients as we could, do whole genome sequencing on those, implant them into mice. Once the tumors grew, sequence the tumors from the mice as well, and then develop cell lines from the PDXs, and then use those, what we call PDX-derived cell lines. So a lot of the work that I'm going to talk to you about today is our insights into osteosarcoma based on these PDX-derived cell lines. So we've analyzed quite a few of these samples since we started this effort. We've sequenced about 98 samples from about 55 patients. We have about almost 50 whole genome RNA sequencing pairs from the same tumor, a significant number of samples from metastasis, and also a significant number of longitudinal samples obtained at different time courses from the same patient. And the little table on the left indicates, we're not the only ones who have done this. There are other efforts out there to do similar things. One of the things we're trying to do is to try to collate all that information and organize it so that we can, have a bigger data set of samples to work from. This was our first effort to do that. It was published a few years ago now. Each one of these columns is a sample. Each one of the rows is a so-called cancer gene we've curated as we think as likely to be relevant to cancer. And really the take-home message here is just how incredibly heterogeneous these tumors are in terms of their likely common drivers. So for example, there's a subset of tumors that have high, the orange and the yellow indicates amplification, the blue indicates deletion. So the second row from the top is MYC. So MYC is one of the more commonly amplified genes. Cyclin E is another one, VEGF-A, but every patient has essentially sort of a unique collection of different amplified regions and also unique deleted regions. The only thing that tends to be pretty common, which maybe isn't surprising for such a complex genome, P53 is almost always altered in osteosarcoma. But one thing that's very unique about osteosarcoma, unlike most cancers where P53 is lost by either point mutation or by deletion, in osteosarcoma, it's almost always structural rearrangement. So there's a break in intron 1 that causes a break in the expression of the gene, and that's how it's almost always deleted. This is a higher magnification view of what I showed you before, just again to highlight the complexity of these tumors in terms of not only numbers of chromosomes, but also the rearrangements in the chromosomes. And chromothripsis, which is a phenomenon where a chromosome basically is thought to shatter in a micronucleus, get reincorporated into the genome. And then so you have these patterns of chromosomal gains and losses on a single chromosome. And that's a very common phenomenon in osteosarcoma. But the other thing which I still find quite fascinating, maybe hard to see, but we asked Betsy Hirsch to do FISH for MYC, because we knew from the whole genome sequencing that this tumor had high amplification of MYC. And if you're familiar with whole genome sequencing, you can get a little bit fooled into thinking that there's an amplification on a particular chromosome because you align those reads to that chromosome. So when you align the whole genome of this patient, you see a big pileup on chromosome 8. You think, oh, well, the MYC is amplified on chromosome 8. But actually, there's MYC signal on every single chromosome in this genome except for chromosome 8. It's basically scattered all over. And so that probably indicates something related to this phenomenon of the chromothripsis, or perhaps ecDNA, which was initially developed as a part of the chromothriptic process. And then that MYC gene was amplified and then essentially reinserted all over the genome. So this just kind of, again, explains why these genomes are so complex. And may also explain a phenomenon that we see quite often, which is that MYC copy number levels don't necessarily correlate with MYC protein expression. And that's probably because the MYC protein expression is going to be dependent on where this gene gets reinserted into the genome. If it gets reinserted right downstream of a high potent enhancer, then you're going to get a ton of transcription off of that locus and a ton of protein. This is just another view that kind of highlights how complex these rearrangements can be. So this is again just the short read whole genome sequencing data, and you see these arcs are basically rearrangements or split reads on the whole genome sequencing that indicate that MYC is which is supposed to be on chromosome 8 is linked to chromosome 20, to chromosome 11, to chromosome 19, and you just see these incredibly complex rearrangements. One thing that's important to keep in mind, though, is that you will often hear that tumors like osteosarcoma that are complex like this, people will use the term chromosomally unstable. Kind of that term gets used a lot. But what we found, at least in osteosarcoma, is that most of these very complex rearrangements that you see here actually probably happened very early in the development of the disease. So when you look at the biopsy, you look at different parts of the biopsy, you'll see the same rearrangements. And if you look at the metastasis, you'll see the same rearrangements. So that suggests that this is kind of a very early event that is probably fixed in the genome. So the genome will continue to evolve, as I'll show you in a minute, but it evolves on the sort of a platform or a structure that's sort of set very early on, and that is probably set because there's only, once you figure out how to be a tumor and how to grow aggressively, you can't sort of change that too much because you're kind of fixed on an evolutionary track, right? Which is why I think that these tumors are probably fixed the way that they are. Although you do see, and we do see when we look at the longitudinal comparisons between, say, an early biopsy and a metastasis, that even though the structural rearrangements are mostly fixed, what does change a lot is the copy number. So the number of chromosomes. Here, what you're seeing on the left here are, and the blue is the minor allele, and the orange is the blue is the major allele, the orange is the minor allele for these comparisons. So each one of these rows is a different pair of an early versus a late tumor. And this just tells you how much of the either one chromosome or the other chromosome has changed in terms of copy number. So this one at the top, this number 454, almost half of the entire genome has a different copy number than it did in the initial time point. Whereas this one at the bottom, it's a much smaller fraction. So it's only 163 megabases. And you can see this example here, 454. This is the early sample from the biopsy, and this is the late sample from the metastasis. And the changes that you see here are regions where the orange and the blue are regions where there's a different copy number between the early and the late sample. So on the kind of background of this fixed structurally rearranged genome, you do acquire or lose additional copies of individual chromosomes. And despite the complexity of that, the heterogeneity that I described before, we do see common either gain sites, like so MYC being one of the more common, cyclin E, as I mentioned, being another one. This is just summary across the entire data set of tumors that we've sequenced, which we've sequenced about 60, I think, at this point, or 60 patients. So we do see common regions of gain and loss, so indicating that this isn't a completely random process, but there are oncogenic events that are selected for and that are likely to be required for these tumors to continue to evolve. So as I mentioned, we've, in addition to the primary tumor samples, we've also developed quite a few of these patient-derived models. So these are tumors that we take from a patient and we graft into a mouse. Then we make cell lines from those mice and study those as well. And so these models have been quite helpful for a variety of studies that I'm going to mention to you. One of the things that convinced us that these models were probably good models to study the disease is that what we did initially was to take generate RNA sequencing from the patient tumor, from the PDX or from the PDX-derived cell line, and compare those three. So when we had the trio from the patient, the PDX, and the cell line, we could compare how similar they were to each other. And we were a bit concerned when we started this that maybe all the patient-derived cell lines would sort of cluster separately on a dendrogram based on gene expression. But what we actually see is that they cluster, I don't know if you can see this very well, but they cluster with the patient. So if a cell line came from a particular patient, the transcription profile will be maintained and will be most similar to the patient from which it came from, which gave us at least some sense of security in using these models to study the human disease. You can see that a little bit better here. We also noticed that a lot of the cell lines that are kind of more established, the ones that I mentioned that you can get from ATCC, like SJSA and MG63.3, tend to cluster separately, which suggested to us that there was a lot of biology that was being missed in those cell lines, and that we were capturing that by expanding this cohort of models that perhaps reflected the disease biology better than cell lines that have been in culture for many, many decades. This is just another view of that. These are the trios that I mentioned, the patient, the tumor, the PDX, and the cell line, kind of all together here. And this is the whole genome sequencing, and you can see that in most cases, the rearrangements that we see in the patient are maintained in the PDX and they're maintained in the cell line, again suggesting that these models are probably pretty good models to study the disease. So to develop that a step further, we've also taken these patient-derived cell line models and developed systems to engraft them back into mice using an orthotopic model where we put the tumor into the hind limb of the mouse. We can monitor development of metastasis either with micro-CT or with bioluminescence. We can then amputate the hind limb. So we have the mice recover very quickly. They run around for several more months, and then we can follow development of metastasis in the lung, which is, as I mentioned earlier, the most common site of disease. And so this allows us to study response to various interventions that hopefully will diminish the development of metastasis over time. So going to switch gears a little bit here to talk about some work that we're very excited about. There was mostly the work of very talented postdoc in the lab, Eunice Lopez, who came to the lab with the idea that, okay, so you've done all this genetic analysis, whole genome sequencing. How about if we take a different approach and we look at the epigenetics of these tumors rather than the genetics? And could that perhaps help us to find some patterns that would get us through some of this incredibly complex rearrangements that I mentioned? So Eunice brought to the lab a technique that we had not used before, but that was developed a few years prior by a colleague at Stanford, Will Greenleaf and Howard Chang, which essentially makes use of the ability of transposons to insert themselves into open regions of DNA. You can tag those transposons with sequences of DNA so that you can tag the regions that where the transposons are integrated. Then you can sequence those regions and basically identify open versus closed chromatin. And as you all are aware, open chromatin is where transcription is actively actually happening, whereas closed chromatin is regions that are not accessible to transcription factors. So this has become a very widely used technique that allows you to get a kind of a snapshot of the chromatin remodeling and the epigenetic state or cell state of a particular tumor. We were initially interested in understanding whether this epigenetic states may contribute to an understanding of metastasis, with the hypothesis that during the metastatic process, different regions of DNA would need to be accessible in order for the tumor to adapt to the metastatic site. But what Eunice found when she first did this on the panel of cell lines that we had developed, both our own patient-derived xenograft lines, cell lines, or commercially available cell lines, was this pattern here, which kind of immediately drew our attention because we had sort of expected, given the heterogeneity that I described at the gene level, at the genome level, that we would just see a scattering of the ataxia kind of throughout this principal component. But what Eunice saw was that there was essentially two clusters. If you just look at the first principal component, which explains 46% of the variance, there's a cluster here on the right, and there's another cluster here on the left. Then we went back and sequenced a larger number of the PDXs themselves. So these are the tumors in the mice that have never touched plastic. And again, we see clear separation into two clusters in principal component 1 and potentially a third cluster if you look at principal component 2. Then in black are actually samples that we were able to obtain from patients. So these are direct tumor samples from patients. Most of them fall into this cluster on the left that we call EC1, but we do see this one, and we have several more now that fall into principal component 2. So that suggested to us that maybe there was this kind of underlying epigenetic dichotomy in osteosarcoma, where some samples were more, kind of in this right-sided cluster that we call EC2, or the left-sided cluster that we call EC1. So this is just a slightly different view where you can take those regions of open chromatin defined by ataxic and identify those that are most differentiating between these two clusters, and then just do a heat map, and you can see here that there are essentially 3 groups of samples. There's this one group here that's more, very clearly has open chromatin regions here. This cluster that has another set of very distinct open chromatin, and then a third cluster that is a little bit mixed, that is kind of in between. And these clusters correlate nicely with several important features of osteosarcoma. So, for example, all of the mixed high copy number samples fall into EC1. EC1 or cluster 1 also has all of the samples that had very highly aggressive phenotype when we studied them in the orthotopic model. But interesting, most of the available cell lines, the ones that have been, as I said, around for many decades, were in cluster 2. So if you just looked at those cell lines and you didn't use these patient-derived lines, you would have probably missed this other cluster. So here's some examples. For example, this is a gene IBSP1 where there's a lot of open chromatin in cluster 1, mostly closed chromatin in cluster 2. This gene is the reverse, so TM4SF1 is open in cluster 2, but not in cluster 1. And that correlates pretty nicely with the differential expression of these genes. So IBSPP1, much higher expressed in cluster 1, much lower expressed in cluster 2, and vice versa for TM4SF1. You can then assign those clusters, those attack peaks, to the most likely regulated gene by just using a very simple heuristic, just assigning them to the most closely, the closest gene, which isn't always the most likely regulated gene, but at least it gives you a sense of patterns of gene expression that are different. And when you do that, looking at the peaks in cluster 1 and peaks in cluster 2, the genes that are associated with those two clusters are very different. So in cluster 1, we see a lot of genes that are associated with organ development. Interestingly, renal development for reasons we're not entirely clear, although that might have something to do with Wnt signaling, as I'll mention in a minute. Whereas cluster 2 is much more upregulated in genes that are involved in cell motility, cell migration, chemotaxis, MAP kinase cascade, which will come up again. The other nice thing what you can do with Taxi is because you have these regions of these peaks of enriched open chromatin, you can then, using several different computational approaches, identify transcription factor binding motifs that are upregulated in or enriched in peaks from either of your two clusters, in this case, cluster 1 and cluster 2. So when we did that, well, it was quite interesting to see that the open regions in cluster 1 tended to be associated with some of the transcription factors that I mentioned to you earlier are involved in bone development, like RUNX2 and RUNX3, whereas the genes in cluster 2 are mostly, for those of you who study RAS biology, these are mostly AP1 transcription factors downstream of MAP kinase signaling, FOSL1, FOSL2, things like that. And that correlates nicely with the, as I said, with the open or closed peaks in the binding sites for these different transcription factors. We can correlate that also at the level of gene expression. So this is just RNA sequencing, looking at the expression of those genes. They tend to sort of be co-regulated. So if you look at a particular gene, sorry, a particular sample has high levels of expression of TEAD1 or FOSL2, it's going to tend to have higher levels of expression of other transcription factors in this cluster, whereas samples in cluster 1 have co-regulated high expression of these genes. A parallel way to look at epigenetic regulation is to look at this H3K27 acetyl mark, which is a histone mark generally associated with open chromatin. And so we wanted to sort of corroborate the ATAC-seq results with a different approach. So we did cut and run for H3K27 acetyl. And interestingly, the coloring here, the orange and the blue coloring, is the same coloring that we had basically assigned using the ATAC data. And it correlates very nicely with the H3K27 acetyl. So if there's an open peak by H3K27 acetyl, there's generally an open peak by ATAC, which isn't terribly surprising, but is nice to see that matching up. Which H3K27 acetyl, you can also look for so-called super enhancers, which are regions of basically large regions of H3K27 acetyl, high binding that are generally associated with very high levels of expression of oncogenic drivers. And each one of these samples tends to have its kind of unique pattern of super enhancer activation based on H3K27 acetyl data. But again, you tend to see much closer similarity between the super enhancer regions in the cluster 1 samples and those in cluster 2. Of course, at the end, you want to, correlate this with protein expression. So Anissa then did Western blotting for some of these key genes that we had identified from the attack data and from other genes that we were interested in. And it correlates extremely nicely with what we would expect to see based on the attacks data. So as I mentioned to you before, MYC is almost much higher expressed generally in cluster 1 compared to cluster 2. RUNX2 is a gene that, as I told you, was one of the genes we identified from these ataxia peaks, almost exclusively expressed in the cluster 1 samples and off in the cluster 2 samples. Phospho1 and Phospho2 are the reverse, so they're up in cluster 2 but down in cluster 1. Interestingly, as I mentioned, RAS is upstream of Phospho1, and so that also is associated with upregulation of phospho-ERK, which is much higher in the cluster 2 samples. Also, total levels of ERK are much higher. And that can also be validated by looking at whether these transcription factors are directly bound to chromatin. So this is just looking at total cell lysates, but if you just if you pull down the chromatin and then ask, is a transcription factor bound to the chromatin, the transcription factors that are identified by ATAC-seq as being bound to the open chromatin are also confirmed by Western blotting. You can see that here where phospho1, for example, is up in these two, highly bound to chromatin in these two samples that have that are in cluster 2, whereas MYC is much more bound to chromatin, and so is RUNX2 and RUNX3 in the samples that correspond to cluster 1. Many of you are probably familiar with this resource, the depth map, which was generated by the Dana-Farber, where they took a large number of cell lines, hundreds of cell lines now, and have done whole genome CRISPR screens to identify cell line-specific vulnerabilities. So they do have some osteosarcoma cell lines, not very many, but there's a few. And so we wanted to ask whether these genes that are upregulated or transcription factors that are upregulated in cluster 1 or cluster 2 are associated with vulnerability to those loss of those transcription factors in the depth map data. And although the data is limited because there's not that many cell lines that we can clearly associate as being either cluster 1 or cluster 2, we generally do see a nice correlation in the dependency of these cell lines to these loss of these transcription factors that are, either cluster 1 or 2 regulated. So this data is actually very reminiscent of work that was published now several years ago in another pediatric cancer called neuroblastoma, where it's been well described by several groups that there are these two subtypes of neuroblastoma, so-called adrenergic and mesenchymal, that are also driven by distinct transcription factor networks. And so we're doing additional work now to try to really tease out these dependencies that are key to the proliferation of these different subtypes of osteosarcoma. But one thing that we've done, we were curious as I kind of set it up at the beginning, that there's a process of normal bone development, and then one of our hypotheses that perhaps these different subtypes of osteosarcoma are due to the fact that maybe these tumors are initiated during a different point in the process of bone development. So that's not an easy question to answer, but we had access to well, in this case, one ATAC-seq sample from a human skeletal early progenitor. So this is a paper that was published by Chuck Chan from Irv Weissman's group a few years ago, where he had actually isolated either early or late bone progenitors from fetal tissue, and then demonstrated that the early bone progenitors, the so-called HSSCs, had more capacity to form the entire subtypes of bone tissue than did the late bone progenitors. So we took these HSCs and we mapped them onto the same. He had shown some attack data in that paper, so we took that attack data, mapped it onto our attack data, and encouragingly found that these HSCs actually mapped together with the other samples that we consider to be part of EC1. In contrast, HFOBs, which are a human fetal osteoblast cell line that is also available from ATCC, which is thought to be likely derived from a later progenitor, is found in this other cluster. So that suggested to us, or at least supported the idea that maybe what we're seeing here are these epigenetic clusters correlate to some extent with different developmental origins of the disease. So the next thing we wanted to do is, well, what is the clinical relevance of this? So what Anissa did was to combining the attack data and the differentially expressed genes in the two clusters from the cell line data that we had generated, generate a gene signature of genes that we consider to be either representative of cluster 1 or representative of cluster 2. And then we map that onto human data. So we have a lot more human RNA-seq data than we have human attack data. So we were able to do this analysis with the RNA-seq, whereas it would have been difficult to do with attack. But what you can see here, so each one of these columns is a different patient. We did see essentially two primary clusters showing up here using this attack-seq generated signature, suggesting that human samples do tend to fall either into EC1 or into EC2. I think I forgot to put it in here, but we've also done some preliminary analysis now showing that these two clusters in the human data do correlate pretty strongly, actually, with prognosis. So samples that fall into EC1 tend to have a worse prognosis. Samples that fall into EC2 have a better prognosis. So this begins to suggest that there's some clinical relevance to this. Yeah, go ahead.
Primary tumor, or it's really they start as two different.
Oh yeah, good question. So for this analysis, we focused only on the biopsy samples because we wanted to show that at diagnosis, this signature was diagnostic, or was prognostic. Interestingly, though, when we look at the metastatic samples, we don't have very many examples because we don't have very many patients where we've been able to look at multiple metastases from the same patient. They generally do correlate. So if a patient has a primary tumor that is from cluster 1, the metastasis will be from cluster 1. But there are actually also some patients that are in cluster. It's not 100%. So there are some patients in cluster 2 that also metastasize, although the tendency tends to be lower. But they also continue to be in the same cluster. Maybe I'll skip this for running out of time. But we've just done some correlation at the histological level to show that the markers of cluster 1 or cluster 2 correlate with protein expression in patient tissues. But what we were more interested in doing is, okay, well, we've identified these two clusters. We think they might have prognostic significance. So they also potentially suggest differential approaches to therapy. So maybe instead of treating all patients with MAP chemotherapy, which, as I suggested, hasn't really changed for 40 years, we need to, using these clusters, we could define new therapeutic approaches. So we had, in parallel to this epigenetic work, done a large screen using the cell lines that you see on the left against the drugs that you see on the top. So this was a cell line drug screen of about 700 combinations, completely agnostic to this idea that there were potentially two clusters. So once we had this data, I asked one of my computational biologists, can you look back and see if there are any drugs that are correlated with better response in cluster 1 or better response in cluster 2? So the way to read this is essentially, this is a complicated figure, but the size of the inner dot indicates the area under the curve. So a small pink dot indicates a drug that is highly effective in that combination. So for example, here, this chemotherapy agent with this cell line gave a good response, whereas this TKI with this cell line basically didn't respond at all because it's a big green dot. So small pink dot, good response, big green dot, not a very good response. So what we found quite interesting was that when you look at this data and look at it this way, in the context of these two clusters, these three drugs clearly had a differential response. So trametinib was very effective in cluster 2, not very effective in cluster 1, so this is the IC50. Whereas these two drugs, which are aurora kinase A and aurora kinase B inhibitors, were very effective in cluster 1, but not in cluster 2. And if you recall from my earlier analysis, cluster 2 was highly upregulated in phospho-ERK. So actually, it was very reassuring to see that these samples are responsive to trametinib, which is an ERK inhibitor. Why the cluster 1 samples are more responsive to aurora kinase A and B, we're not really sure yet, and that's work that's ongoing. So we've done quite a bit of validation. Eunice has shown that these results are indeed true, so that the differential response holds up with multiple other cell lines. Here, for example, you can see that these 2 cell lines, which are in cluster 2, highly responsive to trametinib. This cell line, which is in cluster 1, no response to trametinib. She's then gone on to validate that in these in vivo models. So here's a cell line that we think belongs to EC1 based on the attack profile, responded really nicely to the aurora kinase inhibitor, not very much to trametinib. Here's the reverse. Here's a cell line that responded to trametinib, but not very much to aurora kinase B. Eunice has then gone on to show, and this is a little bit surprising, that actually she gets a better response if she combines both drugs in the same mouse. So if she combines trametinib and aurora kinase B, even though we think they belong to one cluster or another, that's where you actually start to see regression. So we have some preliminary data from single cell analysis that suggests that maybe what's happening is that actually, even though in the bulk data, it looks like the tumor falls into one cluster or another, if you look at the single cell data, there probably is some plasticity, and there's some ability of cell lines to kind of toggle back and forth between these 2 cell states, which would explain why when you treat them with both drugs, and you're actually kind of attacking both cell states at the same time, and you get a better response. So this is kind of the model that we've developed so far, which is that we believe that osteosarcoma probably exists in these 2 distinct cell states, maybe somehow connected to the cell of origin or to some other factor that drives these kind of epigenetic plasticity, and that these epigenetic states are amenable to distinct therapies, and that perhaps if we could figure out how to combine these better, we would have a novel approach to therapy. So our ongoing efforts are really to determine the mechanistic basis for these distinct transcription factor activation states, to identify the core regulatory circuits in these different epigenetic states, and to continue to evaluate intratumoral heterogeneity using single cell approaches. So I just hope to have convinced you that while osteosarcoma is a genomically complex disease, this kind of epigenetic analysis could potentially identify both important aspects of the cell biology and the genomic regulation, but also identify potential novel vulnerabilities to treat these tumors. And so with that, I'd like to just end and thank the members of my group, and particularly Eunice Lopez Fuentes, who did this work, and Alex Lee, who's a very talented computational biologist, as well as Andrew Clugston, who did the majority of this work. Thank you for your attention.
Question. So you mentioned early on that you're looking at I wondered if anybody's really looked at how the different osteosarcomas affect lymphocyte development, specifically B cell development, and whether those niches in the bone marrow and are differentially affected, whether you can use that as a surrogate kind of biomarker for regression.
Yeah, really interesting question. We haven't looked a lot, well, probably because we don't know, we wouldn't know how to do it because we're not immunologists, but we haven't looked a lot at B cell development. But there is now some preliminary, well, I say preliminary because it's published, but it's small data sets of single cell data from osteosarcoma. There's a paper in Nature Communications, and there's another one, I can't remember from what journal. So there's maybe like 13 or 14 primary tumors that have been analyzed by single cell. What you see in the tumors is actually mostly macrophages. There's a really strong infiltration of macrophages. But if I remember correctly, there is some, and I was actually surprised to see this, there is some signal for B cell infiltration, but I don't know of anybody who's really spent any time looking at that. What we've been doing in this, in the context of innate immunity, which I didn't really have time to get into, is looking at the role of the STING pathway, which may be familiar to most of you, but it's it's an ancient cellular viral response pathway that detects the presence of cytosolic DNA, which obviously shouldn't normally be present, but in tumors with complex genomes that have tendency to chromosomal missegregation, you get the presence of these micronuclei, which sometimes can lose their nuclear membrane and then expose DNA to the cytoplasm. And that's a phenomenon that we see quite commonly in osteosarcoma. And so we do see a misregulation of the STING pathway, and we have some interesting preliminary data that we can reactivate the STING pathway and cause an increase in immune activation. But we haven't looked too much at how much of that is B cell dependent, but would be interesting to do that.
Really nice talk. I was curious about why there's this increased susceptibility for osteosarcoma metastasis to the lung. I was wondering if in any of your single cell experiments you saw any changes in like cell adhesion molecules that are.
Yeah, really good question. It's a question that's been in the field for a really long time. And I don't know that we really have an answer for that. I mean, there was a nice paper in Nature Medicine maybe 3 or 4 years ago where they did some comparison of using I think it was mostly H2K27 acetyl of metastatic versus primary tumors. And they did find something that actually was sort of surprising. It wasn't an adherence molecule, but it was something, if I remember correctly, was related to the complement system. So sort of indirectly related to, kind of extracellular processes. But yeah, I think it's something that definitely deserves more attention. It's hard to know how much of that you would really pick up with these kinds of omic approaches. You probably would need to do, ideally, some kind of like protein surface omics or something, right? Because especially surface proteins, the expression at the RNA level tends not to correlate so well. And a lot of these things, as you probably know, are, post-translationally modified, they're glycosylated, all kinds of stuff that it's not going to be reflected in the RNA. But yeah, great question.
I had a question about the EC1, EC2 plasticity that you talked about. Do you have any clues as to whether you think that's like a cell intrinsic plasticity or something that's shaped by microenvironment responses?
Good question. I mean, I think our analysis was mostly sort of cell autonomous, in the sense that we did it on cell lines, but that's not to say that there couldn't be significant plasticity in vivo and the different, kind of niches in a patient could lead to, a plasticity between these two clusters. And it's something that we need to look into more, but I think it's a good question.
Do you have any clues from all of this characterization about what are the changes in the metastases? So the chromosomal structure is similar, but you mentioned that they're very resistant to treatment when they're recurrent or.
Yeah. So why, is that because of some chromatin accessibility? Yeah. It's funny because that was the initial question when we started this work. Then we got kind of distracted or redirected towards this whole kind of therapy response issue. But it's something we need to go back to and look more carefully. I think we also need, what we really need is more, ideally what we'd like to have is more paired samples, attack samples between primary tumor and metastasis from the same patient to really be able to ask your question in a more direct way. There's so much heterogeneity that I'm not sure it's going to be very helpful to look at, say, 10 metastases from various patients and then primaries from different patients. You really need the matched samples because otherwise you're just going to be overwhelmed by the heterogeneity. Yeah. Comparing the, primary to the metastasis. Yeah, in the model. Yeah. Yeah.
About potential therapeutics that still might be effective or not. It would be a big experiment, but you have a nice model to start to think about, okay, what selects for metastatic behavior or.
Yeah, yeah. Yeah, no, absolutely. Anybody wants to move to San Francisco and come and study osteosarcoma metastasis, so do a little advertisement, yeah. That's a good question. I've always been, I'm of the mind that there has to be some prior event, right? Because if it's happening, if there's a structural rearrangement in P53, what allows that structural rearrangement to occur in the first place, right? That wouldn't be tolerated in most cells, right? So there has to be some either a specific vulnerability of cells in the bone environment that are more susceptible to this kind of breakage. Because it's not a point mutation like it is, or deletion. It's a structural rearrangement. So that what allowed that to happen. So we don't know, but it's a good question.
I mean, why do you think there is so much aneuploidy in osteosarcoma cells?
I don't know. I mean, I have a colleague at Dana-Farber, David Pellman, who's a very smart guy, and he's convinced that it has something to do with the sort of tension in the bone and the kind of the matrix probably induces some fragility in the nucleus that then makes it more susceptible to chromothripsis and other events. But he hasn't demonstrated that, and we haven't demonstrated that. So. Yeah, yeah. Yeah, you mean like, differentially expressed? Yeah, really good question. So we've looked at that a little bit. We've looked at like some of the SWI/SNF regulators, like SMARCA1 and I always forget the SMARCA1A and SMARCA1B. And we haven't found anything that is super obvious yet. But we probably need to look a little harder.
Put one more over here.
Yeah.
We're probably out of time.
MDM2 is occasionally amplified, but it's not that common. Yeah. But we do see it sometimes. There's actually an interesting subtype of osteosarcoma called paraosteal osteosarcoma that's very unusual and usually not that aggressive. And those tumors almost always have MDM2 amplification. So that's kind of interesting that they're not as aggressive as the P53. They maybe have a different mechanism of initiation or something, The MDM2 specifically? Not that I'm aware of.